Okay, so what I'd like to do now is review one of the processes by which cells segregate replicated genetic information. There's actually two ways they do this. One is referred to as mitosis, a mitotic cell division, and the other is meiosis, a meiotic cell division. So we're going to focus on mitosis for now. So we're just going to put this in the context of the overall life cycle of a higher eukaryotic organism. We're going to use humans as the example. So again, in this diagram here, all humans start out as a single-celled diploid zygote. That single cell then undergoes many rounds of cell division, specifically mitotic cell divisions, to create multiple copies of the cells. Actually, in an adult, anywhere between 50 to 70 trillion cells within the body. And once those individuals reach maturity, reach sexual maturity, they can reproduce. And so again, specifically with specialized cells within the gonads, the ovaries of females, and the testes of males, there are specialized cell types that can undergo another type of cell division that we call meiosis. And again, meiosis is responsible for producing a very specialized type of cell, a reproductive cell, a sperm on the male side and an egg on the female side. So again, through a fertilization event, these two cells unite, generate a diploid zygote, and the life cycle then continues, the creation of a new individual. So again, these cells that are produced through mitotic cell divisions, again, what we're going to focus on for this discussion, they're responsible primarily, not exclusively, but primarily for producing what we call somatic cells or non-reproductive cells. So again, what are the characteristics of mitosis? So again, before we talk about the details here, I want to put this process of mitosis in the context of the life cycle of a cell itself. So again, the life cycle of a cell, or the cell cycle, is represented in this particular diagram here. And again, what you see is that there's a stage of the cell cycle that we refer to as interphase. So interphase is a non-dividing portion of the life cycle of a cell. The dividing portion of the cell, again, is, in this case, we're discussing mitosis, but in a subsequent discussion, we'll be talking about meiosis. So again, either mitosis or meiosis being the dividing portion of the life cycle of the cell. So you can see that in interphase is actually subdivided into three different substages. There's what we call G1, there's S, and then there's G2. And again, there's different activities that are occurring in the cell in these different interphase stages. So a cell that's in G1 is a mature cell that is carrying out a particular function or activity. So specific metabolic activities that are unique to the particular cell type. So again, a liver cell will be carrying out, or a cell within the liver, that carrying out activities that are unique to the liver. A neuron in brain tissue, again, carrying out activities that are unique to that particular tissue, epidermal cells, skin cells, muscle, and so on. So those cells at that stage of maturity and functionality have the ability to decide whether they want to continue dividing or not. If they make a decision to no longer divide, in other words, remain in an interphase state for a very long period of time, they transition into what we call a G0 stage. And again, so that's again a semi-permanent, non-dividing portion of the life of the cell. So again, when cells enter into G0, a lot of times they will remain permanently in that state and will no longer divide. However, occasionally there are cells that have the ability to transition from the G0 stage back to G1. So after long periods of time, they then make a choice to divide. So once that decision to divide has taken place during G1, the first thing that the cell does to prepare for the cell division is to duplicate or replicate the genetic information within the cell. And again, in a eukaryotic system, specifically the genetic material housed within the chromosomes found within the nucleus of the cell. So again, all of that genetic information is replicated or duplicated during the S phase of the cell cycle. Once that process of duplication is completed, you now have two complete and exact replicas or exactly identical sets of genetic information. You've gone from one copy through the replication to now having two copies of all of the genetic information within that nucleus. And it's that information that is going to be segregated through the mitotic event. So before you can transition to that mitotic event, is that the cell has to transition or pass through what we call a G2 phase. And again, what happens in G2 is metabolic activity that is now preparing the cell for the actual cell division. Okay. So I just want to give you a little bit of an idea as to the duration of these different stages of the cell cycle. So you can see, again, this is, it's tremendously variable, the amount of time that a cell will spend in any portion, any of the substages of interphase and all of the different substages of mitosis. However, sort of an average or a typical representation is given here. So again, you can see the bulk of the life of any given cell is going to be spent in interphase. Okay, so in this example, a total of 16 hours to complete the cell cycle. 15 of the 16 hours are spent in interphase. The longest substage of interphase is spent during the S phase. And again, that's because there's a significant events taking place, replication of all of the genetic information within that cell. So the actual portion of the life cycle of the cell that it spends in this process of dividing, and again, more specifically in this case, a mitotic cell division is 1 hour out of a complete cycle of 16 hours. So very, very a small amount of time overall. So the substages of mitosis, again, in describing this process, cell biologists have subdivided a mitotic event into different substages to make it easier to describe and characterize and so on. So there's what we call a prophase, a metaphase, an anaphase, and a telophase. So again, you can see the bulk of the time during the actual cell division, the cell finds itself in is the end stages at the very beginning. So the prophase, about 36 minutes out of the hour, and telophase, the last stages of the cell division, another 18 minutes. So over 50 minutes of a 60-minute cycle for a mitotic event is spent in those two stages. You can see the other two stages of mitosis, metaphase and anaphase, are very brief, very transient. So again, this is just a typical representation of the amounts of time that a cell will spend in these different stages of the cell cycle. There's actually a tremendous amount of variability from cell type to cell type. So what we have here are representations, actual representations, as visualized under a microscope of the different stages of the cell cycle. So in this example, what we're looking at are cells of a fish blastula. So again, a blastula stage, a very early stage in development, subsequent to a fertilization event where there's lots of mitotic activity occurring and occurring very rapidly. So in any event, up here in the upper left, what we're looking at, we'll just take this cell as an example. You can see the plasma membrane, the cell membrane. You can clearly identify a cytoplasm, and then you can also see the nucleus of the cell. And when you look at that stained nucleus, you can't really recognize any discrete structures. You can't see the chromosomes within that nucleus. So we refer to that as chromatin. So the chromosomes are in there. They're in a very loosely packaged state. And so, again, because of the fact that you can't distinguish individual chromosomes, we refer to that state of the genetic material as chromatin. So then this cell here, this middle representation, again, what has happened is that this cell has transitioned into the beginning stages of mitosis. So more specifically, the first stage of mitosis, which is prophase. So over here, when we're looking at this interphase cell, we can't tell if it's G1, S, or G2, although we know that it's interphase, a non-dividing cell. However, when it takes on this particular configuration, it's clearly evident that something has changed. And what has changed is, again, as the cell has transitioned from the G2 stage of interphase into the early stages of mitosis, and specifically prophase. So how do we know that? What are the characteristics of a prophase? Well, you can see that the nuclear envelope is beginning to break down and dissolve. You can see that the chromosomes are now becoming condensed and compacted so that you can begin to distinguish individual chromosomes within that nucleus. And then lastly, what you see is out here in the cytoplasm, these two darkly staining anchor points, there's two of them, they're producing spindle fibers, these a microtubular network that represents a structure that we call a spindle apparatus. And the spindle apparatus is important for the segregation that occurs through the mitotic event, as we'll talk about. So those are the general characteristics of a prophase, of any mitotic prophase, actually any prophase, whether it be a mitotic cell or a meiotic cell. So then you can transition to the next stage of mitosis. We'll actually go down here to the lower left, which is the better representation. This is what we call metaphase. So again, what are the characteristics of metaphase? Obviously, it looks very different from the prophase stage of mitosis. So what you can see is that the nuclear envelope is completely disappeared. You can see that the chromosomes now are very tightly compacted, and they are aligned along a central axis of this now fully formed spindle apparatus. So it may not be so clear in this diagram, but there's an anchor point here, the centrioles. There's an anchor point over here, the centrioles that were originally found out in the cytoplasm. And you can see this fibrous network that contributes to the formation of the spindle apparatus. So on that spindle apparatus, the chromosomes actually become physically connected to those spindle fibers. And specifically, they become connected through their centromeres. That's the important role of the centromere in cell division. So this is a metaphase. Okay, so a fully formed spindle apparatus, condensed chromosomes aligned along the central axis of that spindle apparatus, and the nuclear envelope has been totally dissolved. So then you transition from metaphase to anaphase. So here's anaphase, a slightly later stage in mitosis. And so what you see here is that those chromosomes that were originally lined along the central axis of the spindle apparatus, some of them have migrated to one pole of the spindle, and one of them has migrated to the other pole of the spindle. So two sets of chromosomes segregating to the two opposite poles of the spindle apparatus. That's what anaphase is. That's actually a very short portion of the overall cycle. It's just the movement from the central position on that spindle apparatus to the poles of the spindle apparatus. So once the anaphase is complete, you have these two collections of chromosomes at the two poles of the spindle. So from that point, you then transition into late mitosis, the last stage, which is telophase. And again, in telophase, that's what we're looking at in this last figure over here to the lower right. And what you see is that now those two collections of chromosomes at the two opposite poles of the spindle apparatus are having a nuclear envelope reformed around them. So that occurs during telophase. Once those chromosomes are enclosed within a newly formed nucleus, is that they decondense back into a chromatin-like state, and you can begin to see that occurring in this figure here. And then lastly, there's no longer a need for the spindle apparatus, so the spindle apparatus has been broken down and dissolved. So those are the late stages of mitosis, and more specifically, events that occur during telophase. However, at the same time, there's another event taking place simultaneous with telophase. So what I've just described, these different stages of mitosis, and what mitosis overall describes, is segregation of replicated genetic information through a cell division. And again, remember, the bulk of that genetic information is in the nucleus of the cell. So it's only talking about or describing the process of or the events that occur in the cell division that involve nuclear contents of the cell. What about the cytoplasm? So the cytoplasm has to be segregated into the two daughter cells as well to complete the cell division. So that's a separate and distinct process that occurs simultaneous with telophase. And this is a process that we call cytokinesis. And so the cytokinesis, again, involves the formation of a plasma membrane that segregates the cytoplasmic contents of the original cell into the two daughter cells. Okay, so this is just what I've tried to do here is just give you, actual representations of these different stages of mitosis, what they look like under a microscope. And so now we're going to look at it, the events of mitosis more schematically, and talk a little bit more in detail about, the genetic consequences of this type of cell division. So here's a summary of the overall process again. Here's a cell that's in interphase. Okay, and then it transitions. Once that cell in interphase makes a decision to divide, it'll transition from G1 to S to G2, and then it'll enter into mitosis. The first stage of mitosis, again, being prophase. We'll skip this one. Late prophase, transition into metaphase, transition into anaphase, and transition into telophase, the last and late stages of a mitotic event. So for this specific example, we're going to use as the representative genome, the organisms, the hypothetical organisms genome, a 2n of 4. So meaning that there's two pairs of homologous chromosomes within the genome. So here's interphase again. Again, interphase, here plasma membrane, nucleus containing this chromatin, again, that represents all 4 chromosomes, but they are very loosely packaged, and therefore they're indistinguishable from each other. So when you stain that nucleus, all you see is this diffusely staining material that we call chromatin. So then out here again in the cytoplasm, as I've described in a previous lecture, is that you have a centriole. And so again, there's a single copy of that centriole. So this is during interphase. Okay, so this cell, again, we don't know if it's G1, S, or G2. Let's assume it's decided to divide. So we're at the late stages of G2 now, and the cell is now ready to enter into prophase of mitosis. So again, what happens during prophase? The nuclear envelope begins to dissolve. The chromosomes begin to condense. And you can see out here in the cytoplasm that centriole has duplicated itself. And those centrioles serve as the 2 anchor points for the spindle apparatus. And so again, those duplicate copies of the original centriole will migrate to opposite sides of the cell and begin to assemble that spindle apparatus. So these are the general characteristics of prophase of mitosis. So then what happens again as you progress further into prophase is you can see now, so this would be called late prophase, or sometimes referred to as pro-metaphase, is that the chromosomes now are fully condensed. The nuclear envelope is fully dissolved, and you can see that the spindle apparatus is nearly fully assembled. So what happens is that once these chromosomes, these condensed chromosomes, attach themselves to the spindle apparatus, they're pushed and pulled until they're aligned along that central plate, that central metaphase plate or central axis of the spindle apparatus itself. Okay, so how do they connect to the spindle apparatus? Well, specifically through their centromeric region. Okay, so that's a very specific role for the centromere found on each of those individual chromosomes. So the last thing I want to point out here is that by late prophase, what you can see is that the chromosomes now are nearly fully condensed or compacted into these very tight structures. And it's at this stage that you can actually see their replicated nature. Remember, these chromosomes duplicated themselves during the S phase of interphase. So that means that, the DNA molecule that ran through the original chromosome, the unreplicated chromosome, the DNA molecule duplicated itself. Okay. So you can see these chromosomes, these replicated chromosomes, those original four, you can recognize the replicated nature. They now have, instead of a linear configuration, they have an X-like configuration. So again, the idea that, again, for a homologous set of chromosomes, the original diploid cell in interphase, there'd be two copies of unreplicated chromosomes. So again, each of these members of this homologous pair would have a DNA molecule running from end to end. After replication during the S phase of the cell cycle, that DNA molecule has replicated itself or duplicated itself, and yet still remain the molecules of DNA still remain within the chromosomes, still remain attached at the centromeric region. So you have these now configurations that are recognized at late prophase and metaphase. You can recognize the duplicated nature of those chromosomes. So these two structures here, replicated chromosomes, are still two chromosomes. But each individual replicated chromosome now consists of what we call two chromatids, and more specifically, two sister chromatids. And the reason we call them sister chromatids is they're exact genetic replicas of each other. Okay, so these are all important concepts. So I've actually shown you these late prophase or metaphase chromosomes in a previous lecture. So again, human chromosomes, here's an electron micrograph of one, and here's another electron micrograph of two human chromosomes at the metaphase, at late prophase or metaphase stage of a mitotic cell division. So again, you can very clearly see the two sister chromatids that are present within each of those individual replicated chromosomes. Okay, so as I said, once those chromosomes attach themselves to the spindle apparatus by late prophase, they are pushed and pulled until they align along the central axis of the spindle apparatus itself. And so this is when you would describe the cell as being in metaphase. Okay, so what's important here is that you can see that each of these replicated chromosomes, remember we started with the genome of a 2N of 4, each of those chromosomes, whether they be homologous or non-homologous, align independently of each other at that metaphase plate. So all four of those chromosomes are aligning independently of all of the other chromosomes at that metaphase plate. So that's an important contrast to things that we'll talk about during a meiotic event. So after they align properly, what happens is you transition from metaphase to anaphase. Actually, what happens during anaphase is now the two sister chromatids that were previously attached within their centromeric region, they become independent of each other. And therefore, during anaphase, you have sister chromatids for each of the four chromosomes segregating to opposite poles of the spindle apparatus. So in other words, collectively, you have identical genetic information segregating to the two opposite poles of the spindle apparatus. Okay, so again, this is an important type of segregation that is distinct from what I will describe when we talk about meiosis. So this is what's called an equational segregation, when you have sister chromatids segregating to opposite poles of the spindle. So that segregation process is pretty quick, and then very rapidly at the completion, when all of these chromosomes aggregate at these two opposite poles of the spindle apparatus, is that you then very rapidly transition into telophase. And again, what happens during telophase? As I said, you rebuild the nuclear envelope around those two aggregates of chromosomes here and here. The chromosomes within the newly formed nuclei will decondense back into a chromatin-like state. And then lastly, the spindle apparatus will break down and dissolve because it's no longer needed. Segregation of replicated genetic information is now complete. However, the cell division is not complete. You still have to segregate cytoplasmic contents of the cell. And so that, again, that's a separate and distinct process from mitosis or telophase. That's what we call cytokinesis. So what happens is that it's a little bit different in plants and animals, but basically the same, is that, you know, you get a pinching off, you get the formation of a newly formed plasma membrane across this region of the dividing cell, and you produce two daughter cells now that contain roughly half the amount of cytoplasm as the original cell that you know, started this process of division. Okay, so that's the completion of the overall process of mitosis. The end goal of a mitotic event, remember, it's mitosis that allows for the construction or the assembly of a fully formed multicellular eukaryotic organism. And the goal of the mitotic type of cell division is to create these non-reproductive somatic cells, all of which contain the exact same genetic information as the original diploid zygote from which those cells are derived. So the idea is to maintain genetic constancy through cell divisions, and that's really the overall goal, the overall outcome of a mitotic event. So again, a little bit different from what you'll see when we talk about and describe the process of meiosis. Meiosis is different in the sense is that there, what you're trying to do is create a very specialized type of cell, one that can be involved in reproduction, in producing an offspring through a reproductive process. So with that, I'll conclude this this summary of mitosis, and then we'll prepare another lecture describing the overall process of meiosis.